So today I'll present the collaborative work about Duchenne muscular dystrophy, and it's a new approach to develop a new disease-modifying therapy for that disease. These are my COI. The presentation, spanning approximately 40 minutes, will cover the topic in 4 parts, starting with an introduction to the disease, followed by the rationale on applying this Neu-REFIX Beta Glucans in DMD, then the preclinical study data, and finally the summary and anticipated future steps. So, as you know, Duchenne Muscular Dystrophy, DMD, is a severe progressive X-linked disorder caused by mutations in the DMD gene, resulting in the absence of functional dystrophin protein. This makes muscle fibers highly susceptible to damage during skeletal muscle contraction. DMD affects approximately 1 in 5,000 male births, with symptoms appearing around age 3. Many patients require wheelchairs by 8 to 10 years of age, and the typical life expectancy is 20 to 30 years, often ending due to cardiorespiratory failure. In DMD, muscle stem cells attempt to regenerate to form new muscle fibers, but the process is incomplete and abnormal due to accompanying inflammation. This cycle of damage and incomplete regeneration leads to muscle necrosis, which is eventually replaced by adipose and connective tissue, causing fibrosis. The prolonged activation of the innate immune response in DMD results in chronic inflammation, further damaging tissues leading to skeletal, cardiac, and respiratory muscles, ultimately leading to death. Importantly, the inflammation must be addressed first. These graphs from the literature indicate the progress since 1970s, when the median life expectancy of those diagnosed with DMD was only 18 years, has increased gradually through 1990s till date from 28 years to 32 years, which is because of both disease-modifying interventions in majority of the patients and in a small group of patients to whom exon-skipping therapies are administered since 2016. Currently, there is no definitive cure for that disease. Therapeutic strategies are mainly of 2 parts. The first approach focuses on restoring dystrophin function through methods such as exon skipping, gene therapy, myosin inhibitors, utrophin modulation, CRISPR-Cas9, suppression of stop codons, and stem cells. However, these dystrophin-targeted therapies have not achieved the desired outcomes. They can slow disease progression, but don't restore the function of abnormal muscle tissues due to the degenerative nature of DMD. Additionally, it is challenging for these therapies to target all muscle tissues, which are widely distributed throughout the body. Moreover, these approaches are expensive. The second therapeutic approach involves disease-modifying therapies that target pathological pathways, including fibrosis, inflammation, loss of calcium homeostasis, oxidative stress, ischemia, and muscle atrophy. This includes steroids, which are used as a standard treatment but have associated side effects. Disease-modifying therapies are gaining more attention because they offer relief from at least some of the symptoms in these patients. Our approach falls under this category, utilizing the potential of biological response modifier, beta glucan called Neu-REFIX, to target inflammation and fibrosis in DMD. From now, I'll share the rationale on why Neu-REFIX was used in DMD as a target disease. So, Neu-REFIX Beta Glucan is an exopolysaccharide produced from a novel N163 strain of black yeast, Oleobasidium polylans, which has been consumed as a food supplement in Japan since 2018. Another strain, A4-202 produced beta glucan, has been consumed as a food supplement in Japan since 1996. These are already consumable and allergen-free beta glucans listed as food additive as per Japanese regulations. The pictures in these slides show at the center the yeast, which is surrounded by the secreted exopolysaccharide beta glucan. In a phased manner, before starting the preclinical mdx animal model studies, a series of studies were undertaken in animal models and healthy volunteers comparing the efficacies of the two variants of beta glucans produced by A4-202 strain and N163 strain, which unraveled the potentials of N163 strain procedure. The Neu-REFIX leading to the discovery of its potentials in DMD. I'll take you through these 4 studies one by one. In the first study, 6-week-old SD rats were used in the study. The groups are mentioned above. Evaluations for blood cell counts, CRP, IL-6, IL-8, IFN, IgA, and immunological ratios such as neutrophil to lymphocyte ratio, NLR, lymphocyte to C reactive protein ratio, LCR, and leukocyte to C reactive protein ratio, LECR, were performed. The Neu-REFIX Beta Glucan enhanced the leukocyte to CRP ratio, shortly called as LECR, which is known to regulate the immune hyperactivation. There was a beneficial increase in LECR in the Neu-REFIX group on day 15 and day 29, shown in the graph. In the second study, 6-week-old male obese and diabetic mouse model, KKAY mice, were used. The groups are mentioned above. Evaluations for biochemical parameters for metabolism, blood glucose, triglycerides, total cholesterol, LDL cholesterol, HDL cholesterol, and NEFA were performed. NEFA levels in male KKAY mice that were already administered Neu-REFIX for 28 days showed a better reduction in group compared to other group and the control. NEFA induces macrophages to secrete inflammatory cytokines. Thus, the entire cascade of metabolic syndrome, diabetes, obesity, and inflammatory state contributing to cancer can be attributed to lipotoxicity. Therefore, individuals may benefit from strategies that target lipids, especially those that normalize NEFA levels, which from anti-inflammatory benefit is of relevance to DMD. Third study was in STAM model of NASH, NASH, mice that are destined to develop liver steatosis and diabetes followed by liver fibrosis were used. Evaluations for plasma biochemistry, liver biochemistry, and histological analysis were performed. The inflammation and fibrosis biomarkers, especially IL-6, was reduced in the NASH animal models where Neu-REFIX was administered. We also evaluated the liver function test and gut microbiome along with metabolome, which were favorable in terms of managing inflammation and fibrosis. Fibrosis, which is the outcome of inflammation and macrophage-associated inflammation, F480 immunostaining, was reduced significantly in the Neu-REFIX group. Among the 5 groups of animals shown in the graph on the left, and the reduction of tissue-level fibrosis to the right. Tubicibacter and Bilophila, the gut microbiome that are pro-inflammatory, showed decrease in Neu-REFIX group, whereas the telmisartan group showed an increase in Bilophila after intervention shown on the left. The study was tested as an exploratory study in 16 healthy Japanese male volunteers aged 40 to 60 years with 4 intervention conditions, 2 test food group and 2 durations of intake in each test food group. The duration of the study, food intake, and the schedule of visit for each group was: Group 1, A4-202 beta glucan, sachet with each meal, so for 35 days or 21 days. Group 2, A4-202 beta glucan, sachet with each meal, so for 35 days or 21 days. CD11B increase, so in the monocyte fraction was observed after 21 days of ingestion, indicating immune activation by Neu-REFIX Beta Glucan. CD11B, an LPS receptor, is involved in bacteriophagocytic activity of immune cells. IgA, D-dimer, galectin, fibrinogen, and ferritin, group 2 in which Neu-REFIX was included showed an increase in IgA levels, along with reductions in D-dimers, galectin, fibrinogen, and ferritin, suggesting the potential of Neu-REFIX in addressing immune inflammation-associated coagulopathy risks. NLR, LCR, LECR in NLR alongside increasing LCR and LECR, which are potential biomarkers of inflammation and hyperactivated immune response, further supports the therapeutic potential of Neu-REFIX in managing diseases with underlying inflammation. To summarize the rationale, the anti-inflammatory and antifibrotic effects at tissue level and plasma biomarkers in animal models of SD rats, NASH and KKY mice, as well as similar potentials in healthy volunteers, made us evaluate a Neu-REFIX Beta Glucan in mdx animal models. Before sharing the data of a preclinical study, I'd like to touch on some basics about Neu-REFIX Beta Glucan candidate on its unique features. There are several beta glucans available in the market derived from various sources, such as mushrooms and yeast. Neu-REFIX beta glucans stand out due to their unique structural formula. Being secreted as an exopolysaccharide, they require no complex purification of refining, resulting in higher biofunctionality. The active ingredient is standardized with drug-like dosage, which is a major strength, making validation easier in preclinical studies. Additionally, it doesn't contain any allergens and is water-soluble, making it easy to mix with water or food and administer orally to the mdx mice. The room temperature storage is another advantage while storing in handling. The picture on the right is that of the factory where this Neu-REFIX is produced, in big bioreactor tanks under GMP conditions, as you see on the right. 2 studies were conducted using mdx mice models. One study focused on the effects of Neu-REFIX on inflammation and fibrosis, examining histopathology and immunohistochemical markers related to skeletal muscle and also myocardial fibrosis along with muscle regeneration. The other study investigated improvements in muscle function by evaluating the fatigue score. This is a study design. C57, so BL10, I call it B10 mice, were used. Group 1 normal, Group 2 vehicle, Group 3 N163 beta glucan were given respectively. The animals were sacrificed on day 45 by exsanguination through the abdominal vena cava under isoflurane anesthesia. Urine, blood, and muscle specimens were collected for evaluation. Quadriceps, gastrocnemius, soleus, plantaris, tibialis, anterior, extensor zygotram lungs, diaphragm, and myocardial muscle tissues were harvested, fixed with extravagant gum, and stored at -80 degrees. The investigations conducted included inflammation and fibrosis assessment through muscle biopsy and histopathological evaluation. Muscle regeneration markers CD44 and MI3 in muscle biopsy and myocardial fibrosis assessment using collagen type 1 immunohistochemistry. Neu-REFIX Beta Glucan normalize the levels of liver enzymes in mdx mice. The Neu-REFIX group showed a significant decrease in the plasma ALT, AST, and LDH levels compared with the vehicle group. Neu-REFIX reduced inflammation in the serum of mdx mice. Plasma IL-13 levels decreased in the N163 group. Neu-REFIX reduced inflammation in the muscle of mdx mice. Representative photomicrographs of hematoxylin and elgin stained muscle sections are shown in the figure, in which the vehicle group showed a significant increase in the inflammation score compared with the normal group. The inflammation score in the Neu-REFIX Beta Glucan group decreased compared with the vehicle group. In muscle trichrome staining, the vehicle group showed a significant increase in the fibrosis area compared with the normal group. The Neu-REFIX Beta Glucan group showed a significant decrease in the fibrosis area compared with the vehicle group, proving the antifibrotic efficacy of the candidate beta glucan. The nuclei of newly regenerated muscle fibers are centrally located, whereas those of mature muscle fibers are peripherally located. In our study, CNF increased in mdx mice compared to that in normal mice due to an increase in necrotic fibers. The number of CNF showed decrease in Neu-REFIX Beta Glucan compared to the vehicle group. We speculate that it could be due to necrosis being tackled by Neu-REFIX Beta Glucan, which we infer by reduction in inflammation score, and also because the number of peripheral nucleated fibers having increased after Neu-REFIX Beta Glucan, showing that normal fibers that are matured are also increased after Neu-REFIX administration. The H score is a commonly used endpoint to assess biomarker expression. The H score algorithm first identifies individual cells, categorizing them as either positive or negative based on the relative expression of the biomarker. Positive cells are further classified into high 3+, medium 2+, or low 1+ categories based on the biomarker's signal strength. The H score is calculated as a weight sum of the proportion of positive cells relative to the total number of detected cells. This score extracts both the intensity and percentage of the biomarker of interest from the IHC image. In the study, significantly higher H score was observed in the Neu-REFIX group compared to groups 1 and 2. CD44 positive myogenic cells isolated from muscle biopsies are known to differentiate into skeletal muscle, both in vitro and in vivo, restoring dystrophin expression and strength. Therefore, the higher intensity of CD44 staining in the muscle biopsies in this study indicates effective muscle differentiation and maturation following regeneration. When calculated using sketch and calc, the MIH3 positive area in group 3, Neu-REFIX treated group, was the highest along with MYH8, which is a gene that encodes embryonic and neonatal myosins, which are highly expressed during early muscle development, downregulated after birth, and re-expressed during muscle regeneration. The 20% higher MIH3 expression observed in this study indicates enhanced muscle regeneration and proper stem cell activation in the Neu-REFIX Beta Glucan treated mice. Collagen type 1, COL1, in the myocardium was evaluated using immunohistochemistry. The percentage of COL1 area in the myocardium was found significantly decreased in the Neu-REFIX group. This reduction in myocardial fibrosis following treatment with N163 beta glucan is particularly noteworthy in the context of genetic muscle structure abnormal diseases such as DMD, where myocardial failure occurs in advanced stages and contribute to premature death. Then I'll move to study 2. So C57BL10, B10 mdx mice were used, group 1 normal, group 2 vehicle, and group 3 N163 beta glucan, so were given respectively. Assessment of fatigue score was performed by measuring the decrement between the first and the last following grip test. The fatigue score decreased in mdx Neu-REFIX compared to that of mdx control group on both day 41 and day 45 significantly, showing that mdx Neu-REFIX mice had better resistance to fatigue compared to mdx control, an indication of an improvement in following grip strength. Neu-REFIX significantly reduced skeletal muscle fibrosis by itself when orally consumed. It enhanced muscle regeneration by increasing MYH3 and CD44 levels. Our study yielding a reduction in myocardial fibrosis with Neu-REFIX in mdx mice needs further validation in clinical settings, as there are not much interventions available currently in late stages of the disease. In the present scenario, where there is no definite treatment available for DMD, and that the current disease-modifying treatments having their own adverse effects, the Neu-REFIX fulfilling safety criteria and efficacies in mdx mice is worth recommending a validation as a safe disease-modifying therapy in long-term studies as an adjuvant standard of care regimen in patients with DMD. Our hope is to extend the lifespan of the patient in safe manner with our approach. As next steps in research, we plan to evaluate the inflammation of diaphragm, quantify IGF-1 and dystrophin levels in specimens stored following the mdx mice study. We are glad to share that last month the Neu-REFIX has been granted orphan drug designation and a rare pediatric disease designation by the US FDA. So hopefully a clinical trial will be started in the United States soon. Thank you so much for your attention. This is the last slide.